Hello everyone, I am Dr. Tilak Maity, Assistant Professor, Department of Biotechnology, Haldia Institute of Technology. Today in this lecture, we are going to explore an interesting and essential topic in plant biotechnology, fundamentals of plant tissue culture. Cell and tissue culture are laboratory techniques used to grow cells or tissues outside of their natural environment under controlled conditions. These methods used to grow and maintain plant or animal cells, tissues or organ in a controlled sterile environment outside the organism. Tissue culture is a powerful tool in biotechnology, agriculture, research, and medicine for studying cellular processes, developing therapies, producing other biological products, conservation, and genetic improvement. Plant tissue culture is a technique used to grow plant cells, tissues, or organs in a controlled sterile condition on a nutrient medium. This method relies on the principle of totipotency, which means that every plant cell has the potential to regenerate into a whole plant. The practice is widely used for the mass propagation of plants, developing genetically identical clones and producing disease-free plants. Plant tissue culture is a technique used to grow and manipulate plant cells, tissues, or organs under controlled sterile conditions. It is based on certain biological principles that allow the propagation, regeneration, and genetic manipulation of plants. The principles of the plant tissue culture are grounded in concepts of totipotency, growth regulation, sterility, and controlled environment. Plant tissue culture has a rich and fascinating history with contribution from several pioneer scientists. The development of plant tissue culture was driven by advances in understanding plant physiology, cell biology, and discovery of plant growth regulators. NOP solution is a nutrient solution originally developed by scientist NOP in 1865 for studying plant nutrition. It was one of the earliest defined solution used for growing plants hydroponically and became a foundational element in plant tissue culture. NOP solution provides essential macronutrients required for plant growth, making it suitable for early experiment in nutrient supply for plants. It has been modified and adapted for various plant tissue culture applications. NOP solution represent a foundational milestone in the study of plant nutrition and early plant tissue culture experiments, while it has largely been replaced by more comprehensive media like MS medium for most tissue culture application. It remains an important tool for understanding plant growth in basic nutrient solutions. In the year 1838 and 39, scientists Sladen and Swan proposed the cell theory, which states that all living organisms are made up of cells and that cells are the basic structural and functional unit of life. This laid the foundation for the idea that individual plant cells could potentially grow independently. Haberland, a German scientist, is often referred to as the father of plant tissue culture. In 1902, Haberland proposed the concept of cell totipotency, the idea that every plant cell has the potential to regenerate into a whole plant. He attempted to culture isolated plant cells from leaves and stems in a nutrient medium but was unsuccessful in achieving cell division. Nevertheless, his theoretical work set the stage for the future advancements. The early 20th century saw significant experimental progress in plant tissue culture. In 1922, Otto Warburg successfully cultured algae in a synthetic medium, demonstrating that plant cells could survive and grow outside their natural environment. Subsequently, in 1934, Philip White achieved continuous growth of tomato root tips in vitro. These experiments confirmed that plant tissue could be sustained in a controlled environment, making a crucial step towards the development of tissue culture techniques. A major turning point in the history of plant tissue culture was the discovery of plant hormones or growth regulators. In 1934, scientists Went identified auxin indole 3 acetic acid, or IAA, a hormone responsible for cell elongation and root formation. This discovery provides researchers with the tools needed to control plant growth in vitro. During 1940s, Smook and Miller identified another critical hormone, cytokinin, which promotes cell division. The combination of auxins and cytokinins allowed scientists to manipulate cell differentiation, leading to the regeneration of shoots and roots from cultured tissues. The development of appropriate nutrient medium was essential for the success of plant tissue culture. In 1962, Murasik and Skook formulated the MS medium. The nutrient-rich medium contains a balanced composition of macronutrients, micronutrients, and vitamin, providing optimum condition for plant growth and differentiation. The MS medium remains one of the most widely used formulation in plant tissue culture today. From the 1980s onwards, plant tissue culture techniques have been integrated with advance in genetic engineering and biotechnology. The history of plant tissue culture is a witness to the power of scientific inquiry and innovation. From the early theories of cell totipotency to the discovery of plant growth regulators and the development of nutrient media, each milestone has contributed to the growth of essential field. Today, plant tissue culture plays a crucial role in addressing challenges in agriculture, biotechnology, and conservation, providing solutions for sustainable food production, plant preservation, and genetic improvement. Totipotency is the ability of a single cell to divide and develop into an entire fully functional organism. In the context of plant biology, this means that a cell from any part of the plant body, whether a leaf, root, or stem, can potentially regenerate into a full functioning plant when given the appropriate conditions. The phenomena is unique to plant cells because they maintain a higher degree of plasticity compared to many animal cells. This remarkable ability highlights the fact that every plant cell remains the complete genetic blueprint necessary to form all parts of the organism. From Haberland early hypothesis to modern tissue culture and genetic engineering techniques, totipotency continues to provide innovative solutions for global challenges in food production, sustainability, and biodiversity conservation. Micropropagation is a revolutionary technique in plant biology that has transformed the way plants are propagated. It involves the rapid multiplication of plants through tissue culture techniques, wherein small sections of plant tissues are cultured in a controlled sterile environment. This method has found significant applications in agriculture, horticulture, forestry, and conservation. Micropropagation allows for the mass propagation of genetically identical disease-free plants. As the global demand for high-quality crops, medicinal plants, and conservation efforts increases, micropropagation will continue to play a vital role in addressing the challenges of sustainable agriculture, horticulture, and plant conservation. Plant tissue culture is a powerful and versatile tool in plant biotechnology and biotechnology, which offering numerous benefits across various fields including agriculture, horticulture, forestry, and conservation. Here are some key reasons why plant tissue culture is used. One of the most significant advantages of plant tissue culture is the ability to quickly produce a large number of genetically identical plants or clones from a small tissue sample. This is particularly useful for plants that are difficult to propagate through traditional methods like seeds or cuttings. Tissue culture allows for plant rapid multiplication, ensuring a consistent and high yield of desired plant varieties in a short time. Tissue culture is particularly effective for producing disease-free plants, especially for species that are prone to viral or fungal infections. Plant tissue culture does not depend on seasonal factors such as temperature, light, or soil conditions. This enables the cultivation of plants in controlled environment, allowing year-round production. Plant tissue culture is a crucial component of genetic engineering and biotechnology. It provides the necessary tools to introduce new genes, into plants enable the development of genetically modified crops with enhanced traits such as disease resistance, pest resistance, improved yield, or stress tolerance. Its ability to overcome environmental conditions, speed up plant propagation, and produce high quality plants that has made in an essential tool in plant biotechnology. There are several types of plant tissue culture techniques. Each type serves a specific purpose, and the choice of technique depends on the desired outcome, whether it is for mass propagation, genetic modification, or research. Here we will explore the major types of plant tissue culture. Micropropagation is the most widely used type of plant tissue culture, particularly in commercial agriculture and horticulture. It involves the rapid production of large number of genetically identical plants from a small piece of tissue taken from a mother plant. Micropropagation is a powerful tool for propagating crops, ornamental plants, and trees on a large scale. Callus culture is a technique in which plant cells or tissues are induced to form an undifferentiated mass of cells known as a callus. The callus is typically formed by culturing small explants such as leaf, stem, or root tissues on a medium containing the balance of auxins and cytokinin. The callus serves as an intermediate step for further differentiation into shoots, roots, and other plants' organs. Organ culture involves the cultivation of intact plants' organs such as shoot, roots, leaves, or flowers under controlled conditions. Protoplast culture is widely used in genetic engineering, somatic hybridization, and development of hybrid plants with superior traits. It also plays an important role in the study of cell biology and plant development at the cellular level. Somatic embryogenesis is a process in which somatic cells are introduced to form embryos. These embryos can develop into whole plants. This type of tissue culture involves the formation of somatic embryos. Anther culture and pollen culture are techniques that involve the cultivation of organs to produce haploid plants. Suspension culture involves the cultivation of plant cells or tissues in a liquid medium rather than on a solid agar medium. Suspension cultures also commonly used in plant biotechnology to produce valuable secondary metabolites. They are also used in genetic engineering to propagate genetically modified cells. Plant tissue culture is a delicate and controlled process that requires precise conditions and techniques for successful plant growth and development. Here are the key points that ensure the success of plant tissue culture. Successful plant tissue culture requires a combination of precise techniques, environmental control, and continuous monitoring of plant growth. Factors such as explant, medium composition, temperature, humidity, and light conditions must be optimized to achieve desired results. An explant is a small piece of plant tissue, organ, or cell that is excised from a mother plant and used to initiate a culture in plant tissue culture. They can be derived from various parts of a plant, including shoot tips, leaf sections, stem segments, roots, flowers, and even somatic cells. Explants are the foundation of plant tissue culture, providing the starting materials for regenerating whole plants under controlled conditions. The success of tissue culture depends heavily on the type of explant chosen as well as the culture medium and the environmental conditions. Whether using shoot tips for clonal propagation, leaves for somatic embryogenesis, or roots for root induction, selecting the right explant is crucial for achieving the desired outcomes in plant regeneration, research, and commercial production. Light plays a crucial role in plant tissue culture, influencing various physiological processes such as photosynthesis, growth, and development. In vitro culture involves growing plant cells, tissue, or organ in a controlled sterile environment, often under artificial lighting. The presence and management of light in plant tissue culture system are essential for ensuring optimal plant growth and successful regeneration. Here are the key reasons why light is required during in vitro culture. Photosynthesis. plant growth and development, control of plant morphogenesis, photoperiod sensitivity, stomatal aperture, and synthesis of phytohormones. Both chlorophyll biosynthesis and photosynthesis are critical processes for plant growth and both require light as a key factor. The amount and type of light significantly affect these processes as plant depends on light to produce the energy required for their development and survival. The ideal temperature and humidity conditions for plant tissue culture are crucial for successful growth and development of plant tissues in vitro. These conditions support optimal cellular activity, regeneration, and differentiation. The optimal temperature for most plant tissue culture is typically between 23 degrees centigrade to 27 degrees centigrade. Some species may require slightly higher or lower temperature depending on their needs. The ideal humidity for plant tissue culture is typically 60 to 70%. These ensure that the plantlets do not dry out during culture while still allowing proper gas exchange in the culture vessel. Both temperature and humidity should be carefully monitored and controlled within the tissue culture laboratory to ensure successful plant growth and development. Adjustments may be needed based on specific plant species and the stage of the tissue culture. The selection of the right culture medium is essential for the success of tissue culture. Murasic and Skog medium is the most commonly used, but other media formulations may be used depending on the plant species. The medium should contain the correct balance of the macronutrients, micronutrients, and carbon sources, which support plant cell growth and development. Vitamins like thiamine and nicotinic acid, along with plant growth regulators like auxin, cytokinins, gibberellins are important to promote cell division, shoot initiation, root formation, and other developmental processes. A well-organized plant tissue culture laboratory is vital for successful propagating plants and conducting research on plant growth and development. The laboratory layout should separate sterile and non-sterile areas, provide control environments for optimal plant growth, and ensure proper sterilization and safety protocols. With the right equipment, sterile techniques, and safety measures in place, a plant tissue culture laboratory can effectively support plant research, propagation, and genetic studies. In conclusion, plant tissue culture is a versatile and powerful tool in modern plant science with broad applications in agriculture, horticulture, and conservation. As technology advances, the potential uses of tissue culture are expanding, helping address global challenges like food security and biodiversity conservation. Thank you for your attention.